Dear colleagues and friends, welcome to the UENPS Hippocrates webinars. And I leave immediately the floor to Professor Noe and Professor Carnelli, who are the organizers of this very beautiful series on nutrition. Please, Virgilio, please, John.
Thank you, Professor Moretti. It's my honor to present our next speaker, Professor Umberto Simeoni, who is in the Faculty of Biology and Medicine at the University of Lausanne and Director of the Division of Pediatrics and of Developmental Origins of Health and Disease, the DOHAD Research Unit at CHUV University Hospital in Lausanne, Switzerland. Professor Simeoni started his academic career at the University Hospital and University of Strasbourg, France, as Director of Neonatal and Pediatric Intensive Care up to 2001. He moved afterward to head of the Perinatal Department at Marseille University Hospital and Marseille University. His research is oriented toward the developmental origins of health and disease, with a focus on the developmental programming of the cardiovascular and metabolic systems under the influence of early nutrition and its epigenetic mechanisms. Umberto Simeoni has served as president of the European Association of Perinatal Medicine, the French Society of Neonatal Research, the French Perinatal Society, and the French Language DOHAD Society. He is currently chairing the Board of Scientific Advisors of the International Foundation for Preconception, Maternal, Fetal, and Infant Health. Good morning, good afternoon, good evening to all of our participants. And Professor Simeoni, you have the stage.
Thank you very much, Joseph, for this kind introduction. And thank you, Professor Moretti, Professor Carnelli, as well, for your invitation. I hope you can see my slides.
Yes, we do.
Okay, so my presentation will be about early events and their long-term consequences, and with a special focus on epigenetic mechanisms. Early development is characterized by a unique particularity. A stimulus from any component of the environment applied during this critical period of early life exerts not only short-term, but also lifelong effects, while the same stimulus applied later has only transient effects. This finding has been named developmental programming, or better, in my opinion, developmental conditioning, and it influences health and the risk for disease over the life course. This is true in particular for nutritional stimuli from the environment, which are shown here in this pioneering work in the 1960s by Widowson and McCann. This is the growth of rats, of normal rats, and a very simple intervention, which is applying a period of undernutrition during only 3 weeks during growth of these rat pups. In this part of the graph on the right, undernutrition was applied between 9 to 12 weeks in the middle of the growth of these animals. And during undernutrition, they lost weight, and then they developed catch-up growth and regained their normal growth with no permanent effect. But the same stimulus applied earlier, between 3 and 6 weeks of life, had an effect of slowing growth, and when nutrition became normal again, the rats started growing, but in a different corridor. They were kind of reprogrammed, and this was for life. So this is a demonstration that it is a time period which is important in this concept, independently on the cause, which is of an intervention. If we look at the graph on the right, this is the prevalence of hypertension in the adult population in a Finnish cohort. And we can measure here the effect of birth weight compared to the effect of current weight in these adults on the prevalence of hypertension. So we hear the effect of perinatal nutrition, and especially intrauterine nutrition, which is simply reflected by birth weight, is extremely important. In addition to the effects, of course, of having a higher adiposity at adulthood. So this graph shows us that subtle variations in initial conditions, as the birth weights which are represented in this graph are almost all within the range, the normal range. Subtle variations can induce very important changes in prevalence of chronic disease at adulthood. We should keep this in mind, and also take into account that later we understood that there is not only a direct correlation, but the correlation has a U shape, which means that higher birth weights have a similar effect of increasing hypertension. And the same could be shown for metabolic diseases such as diabetes, for example. This led to the concept of the developmental origins of health and disease, which means that the common diseases at adulthood, the chronic diseases, which are the most important cause of mortality, of morbidity, and which have a tremendous economic impact worldwide, have early origins. And it is a kind of, I would not say programming, but rather conditioning, because there is no absolute determinism. There is an initial condition which will increase the risk for later disease only from a statistical point of view, and secondary hits later in life can realize this risk and translate it into real disease. An example of this is the effects of the famine, which was observed in the Netherlands during World War II, and a whole region of the Netherlands was submitted during a winter between 1944 and 1945 with a severe restriction to the access to food, which is represented here in the B part of the graph. We have the caloric intake of these people during this period and the external temperature during the winter. We know today that the offspring, which was conceived when a pregnancy occurred during this period, has an increased risk for chronic diseases such as obesity, dyslipidemia, type 2 diabetes, even neuropsychiatric disorders and others at adulthood. And this is running, including in the following generation. So this is a similar observation than the ones I showed in my first slide. And interestingly, comparing the epigenetic profile in terms of DNA methylation, which is an epigenetic change in the surroundings of the genes, considering six genes involved in metabolic diseases, there was a difference in the degree of methylation rate of these genes, comparing one baby who was exposed to this changes according to these nutritional changes in the mother, according to the time of the last menstrual period, and a sibling who had not been exposed. And these changes correlate with the intensity of the famine very nicely in the lower part of the graph. So this shows us that the link between early life events and their long-term consequences is likely to involve epigenetic imprinting, aside possibly other mechanisms, of course, such as the influence of the microbiome, for example, or genetic variance combination. It is certainly a multifactorial phenomenon, but epigenetics seem involved in. About epigenetics, it's simply a molecular translation of early interactions between the genome and the environment. In the absence of any change in gene sequence. This imprinting is obtained through three main mechanisms, which is DNA methylation. This is simply the addition of methyl groups to the cytosine nucleotides in the DNA. Second mechanism, changes in the histones configurations. So histones are the proteins around which the DNA is disposed in the nucleus. And of course, changing the configurations of the histones just changes the chromatin and the accessibility of DNA to, for example, transcription factors. And the third mechanism is non-coding RNAs, which are the major part of our total RNA. And these are regulating RNAs, which act as the two other mechanisms by simply turning on the genes and upregulating their activity, or to the contrary, downregulating it. So epigenetics act in combination with the genome. It's a way of reading the genome. It's like a music which is written, but the interpretation can be performed in an infinity of different ways. And this is allowed by epigenetics. Epigenetics introduces us a huge part of biological diversity, which could not be explained by the genome alone. We have only 20,000 genes which work to build and to regulate our body. And indeed, epigenetics and epigenetic imprinting are important simply because they regulate the function of these genes. It's not only an on-off mechanism, but a fine-tuning. And this fine-tuning is acquired early in life and is connected to the cues received from the environment. Nutrition, for example, but also stress, exposure to toxicants and chemicals, and social vulnerability. All these factors can alter the early epigenetic imprinting, which is necessary to the fine-tuning of development and to the setting of the reference points of the main regulations in the organism. For example, the glycemia or the blood pressure of an individual, which is a normal range, but the precise reference point depends on these early signals from the environment received during early development and translated into epigenetic imprinting. Epigenetic marks are long-lasting and can be transmitted over several generations, so they are inheritable. Although there is no mutation in the gene, no change in the gene sequence. So we can summarize this with this scheme. Early environment constitutes an epigenome, which comes in addition to the genome. The genome is fixed, and epigenetic marks are flexible and dynamic over the life course, and they are archiving environmental impacts, and they up or down regulate gene expression, which then leads to proteins, of course, to the development of organs and systems, their structure and function, and finally, the phenotype of the individual. So this is developmental plasticity, which is that one genome can result into an infinity of possible phenotypes. And we have today extraordinary tools to characterize both the genome with the next generation sequencing, but also the epigenome and other omics, the microbiome, metabolome, proteome, and also upwards the exposome, which is the global exposures to which the individual is submitted. What is important is to understand that development is an epigenetic phenomenon as well as a genetic one. And development occurs in adaptation, in good harmony with the environment, because it's like if the baby was developing according to the signals received by the environment, the early environment, just to be adapted to fit better with this environment over its entire life. So this phenomenon is kind of predictive. And if the early signals by the environment are optimal, are healthy, so the programming of the functioning of the genes or the conditioning will be optimal. And this individual will experience a life course in good health. To the contrary, if the early signals are not optimal due to stress or altered nutrition, even subtle changes, as we have seen, then the consequences can be a dysregulation which persists up to adulthood, is a long time silent, and then reveals by an increased risk of disease. So this time period of early life is important simply because development is at stake, and development involves epigenetics, and epigenetics involve the environment. This is true for the development of the brain. It's this very nice integrative studies which showed the spatio-temporal dynamics of brain development from the embryonic period to adulthood, and the changes in epigenetic modifications, which are very nicely organized in the different regions of the brain and over time during development. And so they can produce an optimal structure and function, or a suboptimal one with an increased risk, for example, of neuropsychiatric disorders, depending on these epigenetic-led trajectories. Of course, when a stem cell commits itself and differentiates, for example, into a neuron or a glial cell, as all the cells in the body have the entire genome, this commitment is allowed by epigenetics, which silences the parts of the genome which are not necessary to the specialization of these cells and enhances the activity of those genes which are completely necessary to function as a neuron. We have also, it is interesting to know that development itself is epigenetic in nature, and it is possible using a panel of genes to design an epigenetic clock, which is predictive, for example, of gestational age, according to the status of the global methylation of DNA in the white blood cells in the blood. And methylation increases during gestation as a general basic phenomenon during development. Also, epigenetics have a complementary role as a memory between a molecular memory between early life environment and long-term health with the microbiome, which is an extremely important actor as well through its interactions with the gut immune system and the generation of possibly low-grade inflammation, also its own metabolic activity and characteristics. And we know today, I have no time to go into details into this, that the microbiome exerts its effects, at least in some pathways, through the means of an epigenetic imprinting of the targets which are affected, for example, the gut immune system or the gut epithelial cells of the intestinal microbiota. So finally, we can understand the trajectories of health over the life course, following the example of these curves, which are the green one is a favorable one for good health, and the red one is a less favorable one, depending on the early environment factors, which affect the slope, the initial slope of these trajectories. Also, naturally, later stresses or exposures to adversity will influence the shape of these trajectories, but the baseline will follow a different slope depending on even very subtle variations in early environments. This reminds me, for those of you who have assisted to the talk yesterday by Professor Fuchs, the early start, which it is necessary to optimize in the growth of preterm infants if we want to avoid extrauterine growth restriction. The first times of life from preconception up to the second year of life as a general frame are the most important because small changes in these slopes translate into very important changes in health at adulthood, depending on the genome, of course, and the epigenome. This is, again, developmental plasticity, which is the highest at the beginning of life. We then have the life cycle, the life course, which is influenced by early environment. The memory, the molecular memory, seems to be, at least in part, epigenetic. And then health at adulthood can be influenced depending on such early environment. But interestingly, the epigenetic imprinting can be inherited by the next generation. And this susceptibility to disease, this risk factors can be transmitted either by the mother or the father, including before the baby is conceived, because the gametes of both the father and the mother can have epigenetic marks which reflect the lifestyle and the environmental exposures of the father or the mother. So this is, of course, something which can be worrying, but it is also a wonderful opportunity, because if it is possible to optimize early environmental exposures, including in young adults who have the project of having a baby, then trying to have a healthy environment and a healthy lifestyle will simply help not only themselves for improving their health, but also the possibility of improving the future health of the baby is open. This can also explain why these chronic diseases such as obesity, hypertension are spreading so fastly across the world today. It's clearly pandemic, even with much higher consequences than the COVID pandemia, unfortunately, because these diseases kill softly. And it is possible that this mechanism of transgeneration, inheritability of what are acquired traits, acquired epigenetic imprints, can have a leverage effect in the incidence and prevalence of chronic diseases in the general population. Now, this is a general figure of the concept, but let's examine what the great perinatal syndromes, which are, for example, intrauterine growth restriction, preterm birth, or exposure to maternal overweight or to maternal diabetes, what these syndromes tell us about this story and the babies who are born after such exposures or with such exposures. The question is this general phenomenon kind of amplified or by these adverse contexts of pregnancy, of conception, and of the neonatal, affecting the neonatal period? What we know is that in very low birth weight infants, when they become adults, they have an increased blood pressure. This is a little increase at young adulthood, several millimeters mercury, but we know today that a few millimeters mercury higher blood pressure at young adulthood translates in a significantly increased risk later in life for developing, for example, coronary artery insufficiency or stroke. And this meta-analysis of several cohort studies is very clear about this. A low birth weight leads to an increased arterial pressure at adulthood in terms of mean arterial blood pressure, but also it's not only a few individuals which have this risk, it's a general phenomenon, a statistical observation with a wide distribution, but the medians and the mean are clearly increased. Intrauterine growth restriction indeed has been associated in observational studies to a number of later consequences. Cardiovascular disease and metabolic syndrome, type 2 diabetes are the more obesity in some cases are well known, but other disorders have been found significantly associated with early exposure to fetal growth restriction, neurologic disorders, respiratory disorders, immune dysfunction, osteoporosis, but also the context with and behavior and mental health and social health, which can be altered, increased risk of depression, renal disorders, and polycystic ovaries disorder as well, and some cancers, especially those which are hormone dependent. If we consider now preterm birth, it is very similar. It's very difficult to make differences to know which one is worse, or if there are specific consequences to preterm birth or to fetal growth restriction. But similar observations have been made with effects on the cardiovascular, metabolic, renal function, respiratory, etc., very similar ones. And it is important to keep in mind that babies born very preterm, about 30% of them have also suffered fetal growth restriction. Finally, on the other side, this is fetal growth restriction is an example of reduced nutrition in utero, nutritional deficiency, early nutritional deficiency, but early overnutrition, exposure to overnutrition, as it happens when a baby is exposed to maternal overweight or obesity or excess gestational weight gain during pregnancy. We have a robust evidence that very similarly, and this can be surprising, I think this is a contrary initial stimulus. The risk for developing obesity in childhood, high blood pressure, cardiovascular complications, type 2 diabetes is increased in the offspring. And there is emerging evidence that other disorders are affected as well, especially if diabetes, especially diabetes in pregnancy, is superimposed over overweight or obesity. Having GDM, gestational diabetes, which is represented here by the triangles compared to the full discs, which simply represent categories of maternal BMI, which means adiposity. Diabetes adds its effects on the ones of maternal obesity or simply overweight. And it is absolutely striking that these great obstetrical syndromes in intrauterine growth restriction, preterm birth on the deficiency side, low birth weight side, or antenatal exposure to overweight or hyperglycemia in pregnancy, despite being so different and opposed, have very similar long-term possible complications in offspring when it reaches adulthood. They have also common mechanisms of programming, which are both structural and functional. This is about hypertension and cardiovascular disease with very similar factors, which we know. A reduced nephron number endowment, which is found both in fetal growth restriction and in macrosomic fetuses and newborns, which can be surprising. Altered responses to stress, altered endothelial function, which is very important for vascular function and development, with an impaired angiogenesis or vasculogenesis in both situations, in all situations, altered endothelium-dependent vasodilating capacity as well, increased arterial stiffness or intima media thickness when it is measured on the carotid artery by ultrasounds, myocardial dysfunction, and the effects of suboptimal postnatal nutrition. This is for the cardiovascular effects, which are similar in nature and in mechanisms. Metabolic disorders as well, with impaired glucose tolerance, for example, and the risk which is increasing for developing type 2 diabetes at adulthood due to a reduced insulin sensitivity, insufficient insulin production on the other side, obesity, and the effects of suboptimal postnatal nutrition as well. So Again, a very different initial conditions, but similar mechanisms and consequences on the long term, which is striking and suggests that there is a common mechanism at the molecular level somewhere, which may explain this similarity between the different organs and systems. And I will show you also between species. And that's the great obstetrical syndromes. They tell us a lot about this concept of the influence of early environment on the development and the early origins of our health or disease. And they seem to be unfortunately, kind of an experiment in nature which amplifies the findings in the general populations. Also because these conditions lead usually to, of course, significantly increased perinatal stress, whatever the initial mechanism. If we have just a look at this molecular mechanism, we can see that they are also similar. And if we go first to preterm birth and IUGR, the best example, I think, is the effect of preterm birth, and we will see that fetal growth restriction has similar effects on angiogenesis, which is altered. And this may explain later hypertension, because if you have a less developed vascular bed, you can, of course, have increased arterial resistance as an endowment at the beginning of life, which translates later into hypertension. To study this, it's interesting to study the endothelium, which is a part of the vasculature, which is responsible for vessels development and growth, and especially the progenitor cells, endothelial progenitor cells, which can be measured in the blood, their quantity and their activity, their capacity to build new vessels. In this experiment, we compared in humans endothelial progenitor cells from the cord blood of control term newborns in this part of the figure, and on the left of babies with a low birth weight, mainly due to preterm birth. And you can see that when transplanting into nude mice, these progenitor cells, if they provene from a healthy newborn, term newborn, they can build a nice new vessel. If they provene from a low birth weight infant, they build an abnormal vessel. So their capacity to build new vessels is reduced. I'm afraid we have no time to describe the other mechanisms which are involved, but we observed an important point is that this alteration in the angiogenic capacity in low birth weight infants correlates with, surprisingly, an accelerated cell senescence of these endothelial progenitor cells. And so preterm infants have paradoxically progenitor cells which are at risk of accelerated senescence. And this may be a key mechanism, and it could be related to a deficit in sirtuin-1, which is a protein which controls cell senescence. And this deficit was found related to changes in the histone 3 configuration, which can explain by an epigenetic mark why these cells moved into an accelerated senescence. Interestingly, we found this as well in animal models, here in rat model of intrauterine growth restriction, altered vasculogenesis, deficit in sirtuin-1 at adulthood. So this is a persisting effect. And in perinatal overnutrition, we move here from preterm and birth and fetal growth restriction to exposure to the contrary, to early overnutrition. The group of Ingram has shown very similar alteration in endothelial progenitor cells numbers and function in situations of exposure to maternal gestational diabetes in utero. So just the contrary, similar angiogenic alterations. And we found, again in mice, this time in an animal model, that simply by providing a transient overnutrition postnatally to mice pups, by reducing the number of mice in the litter, so with access to a higher quantity of breast milk, and only during lactation, 2 or 3 weeks postnatally, at adulthood, this leads to a fatty liver when these mice become adults. And again, we found an accelerated cell senescence, a stress-induced cell senescence, and a deficit in sirtuin-1, which is corrected when reversed, when a caloric restriction is applied at adulthood. Also, a model of high-fat diet given to the mother between gestation and during lactation has direct consequences to the heart, inducing myocardial hypertrophy and sclerosis. And we observed that this is associated with an increased expression of several microRNAs, which are non-coding RNAs, one of the mechanisms of the epigenetic mechanisms involved, especially those microRNA which favor the production of TGF-beta with a change in the microRNA biosynthesis machinery, which explain this observation. Of course, exposure to postnatal overnutrition or intrauterine overnutrition has also effects on the brain. And this is the key of changes in the appetite and feeding behaviors, as intrauterine maternal overnutrition leads to increased birth weight and an increased transfer of all nutrients to the fetus through the placenta, eventually through breast milk, which can induce a resistance to leptin. Thus, an altered appetite regulation, altered fat metabolism also, and high neonatal adiposity, which predisposes to adult BMI increases. So this can act through the regulation of appetite, but also through the central circuitry of for reward, which explains that if the baby is exposed to a significantly higher quantity of palatable elements, such as elements rich in lipids or in added sugars, this preference can persist later and influence the behavior of the baby. I will finish with the role of stress and this extremely nice study, which is in mice performed by our colleagues in Zurich. This model is a model of early stress, maternal stress, and pup stress by unpredictable separation from the mother in the neonatal period, and added to various ways to induce a maternal stress. This leads to disturbed behaviors in the offspring with depression and a reduced acceptance of risk. But as always, or almost always, in conditions of stress, the neurologic and psychic effects are accompanied by metabolic effects, and in particular by glucose intolerance. So this is a common finding in these situations, which likely also to explain how in humans it may happen that physical diseases and metabolic diseases can be associated with mental disorders or social vulnerability as well. And this study has shown that in male offspring, when they become themselves father, They transmit in the sperm some microRNAs which induce the same disorders in their progeny. And by injecting one of these microRNAs, regulating RNAs, epigenetic RNAs, into normal oocytes, you obtain very similar behaviors and glucose intolerance when the offspring becomes adult. So this stresses also the role of the father, not only the mother, in developmental programming. And we have to integrate this, for example, when we consider how to address these problems in the clinic and have a preventative approach of whether in the case of a birth after maternal, exposed to maternal overweight or obesity or diabetes, or also in the general population in minor forms, the preconception period is extremely important. The conception is here in this graph. And it concerns both the mother and the father, their lifestyle, their exposures to stress, to toxicant, to the environment. And through various mechanisms we have described briefly, they can transmit epigenetic marks which can be inherited by the fetus, which is then exposed in utero to the similar environment. And after birth, to the lifestyle adopted by the family and the behaviors. Mimicking the behaviors is the most common way of learning for a child. So both the mother and the father can influence this kind of outcomes and repetition of disorders across the generations, and both should be addressed very early, even in preconceptional programs, to try to bridge these problems. In conclusion, in the perinatal syndromes, what can be done? Sorry. Certainly, in the neonatal period, try to optimize very early the beginning of postnatal nutrition. This is true both in the case of preterm birth or fetal growth restricted infants or macrosomia. But the mechanisms are so complex and the situations are so different that personalization makes sense and is likely to be preferable to a one-fit-to-all approach in the cookbook, as mentioned by Chris Fuchs yesterday, and control that the nutritional intakes are optimized. We have little evidence about the best postnatal strategy, whether we should have an intensive nutrition in low birth weight infants or a more cautious one. Of course, it's a period of intense development of the brain, and this is a priority. I guess simply optimizing, trying to meet the requirements and to adapt to individual situations is the best way, and control, audit that the nutritional intakes, that the requirements are met really, and not only thought in our minds or prescribed. And also, we should certainly try to reduce as far as possible, anyway, extrauterine growth restriction observed in these cases. Of course, control exposures to stressing and toxic environments, and follow up. While we monitor growth and neurodevelopment, we should be aware that other factors, which could be early biomarkers for chronic disease, should be certainly followed up, such as blood pressure, microalbuminuria, adiposity during growth of these infants, and try to promote a healthy environment and family lifestyle. And simply, very quickly, two examples of what can be done in the general population, not only those who are disorders during pregnancy medically characterized. This is an example of what an insurance company has set up to offer a preconceptional early prevention chronic diseases for the future parents and for the offspring, the next generation, through a preconceptional medical visit and personalized e-health and phone coaching, followed on during pregnancy and postnatally, including breastfeeding, even in let's say, normal, if normality exists, people and healthy context to try to optimize the future health of these adults and have possibly a populational effect. And this is in French, I'm sorry, but it's a very recently adopted program for the first 1,000 days in France, which is a set of measures by the French government to optimize the context of this time between conception and the second year of life, especially from the social, psychological, and educational points of view. And I end my talk with these two possibilities of action. Thanking you very much for your attention.
Thank you, Umberto. That was a fascinating lecture. And I think that this is an area that we still have so much to learn. I'm just fascinated by this. Let me ask you a question about, we have people who are now looking at their genomes and looking at the sequence of their genomes, and the cost of that has come down, and people are beginning to act on the sequence of their genomes. So, for example, if they're at high risk for breast cancer, they may get a mastectomy. Now, what about the epigenome? You were talking about the interventions you could have in the first 1,000 days and, having that healthy lifestyle in the first 1,000 days. But sometimes the damage is already done. Mm-hmm. And can we use epigenomic markers when an individual is older, 15, 20, 30, 40, 50 years old, and then intervene so that we can reverse some of these epigenetic effects that occurred early on?
Yes, it's a very interesting question. I would be optimistic and say yes, because, for example, in our experience, we can both reverse a phenotype, which is a suboptimal phenotype, for example, in the context, let's say, on overnutrition, early overnutrition, which leads, at least in animal models, to an altered myocardial function. And although these animals are not obese at adulthood, a caloric restriction, about 20% of energy less in the diet, can normalize the myocardial function. And this is accompanied by changes in the corresponding profile of microRNAs, which were induced early and changed later. So I would say that the epigenetic, the epigenome, the changes in the epigenome are reversible per se biochemically. Of course, these are reversible changes, contrary to a mutation in the genome, for example. But if there are consequences, have been structural, for example, a reduced number of nephrons in the kidneys, then of course it can be a problem. But we know very little, I must say, at the moment. And it's an extremely complex field. The genome is already complex. And we use now polygenic risk scores and the genome has an importance, of course, as well. Maybe more in monogenic or polygenic disorders, such as some breast cancers you mentioned, and in rare disease. But the diversity introduced by the epigenome, which is an infinity of possibilities of tuning up or down all the genes, every gene, of course, to find the right approach and the right target and have an agent which can target this point is extremely difficult. we know that folic acid can have positive effects as a general modifier of methylation, but it's a blind approach, which can be effective anyway.
Thank you, Umberto.
Using
Let me
some-
ask you. Sorry, go ahead.
But using some of these potential biomarkers might be beneficial in terms of targeting certain approaches, and I think that that's really what you're saying, is that it is
Yes,
possible to do that.
It is possible in animal models, and at least theoretically. I must say we are far away from intervening with these agents, but microRNAs are used. For the moment, we use messenger RNAs in the vaccines against the COVID, but they are already used in oncology, for example, for treatments. So maybe I think the microRNAs and the non-coding RNAs are the first field which could be targeted amongst the epigenetic mechanisms for clinical applications, possibly one day.
Thank you.
Umberto, let me ask you, this epigenetic hit, do you think it causes an alteration of the whole body? Is like you see it like a generalized disease, and we measure one parameter at a time because our laboratory we can not measure all, all the parameters. Do you see it as a systemic disease, or you have data, or there are data out there in the literature that sort of strongly support the epigenetic more selectively affect like a cellular line compared to, a structural, is like a more focal disease or is a general disease? I mean,
well, a very important question as well. I try to answer briefly, but I think the mechanism is very general, and it affects all the systems in all the species everywhere it has been studied. It's at work because it works for development, normal development. So it's normal life. Epigenetics and changes is normal life. Then when you have adverse influences in the environment, of course, depending on the profile of this exposome, of the total exposures, there is rarely only one exposure. It's a set of exposures with differing balances between factors, and so they have differing expressions and phenotypes, which results in different risks for differing organs or systems. And it's a matter of combinations between the infinity of possible epigenomes, of timing, of cocktail effects, if we think to endocrine disrupting agents, and of the genome, the underlying genome, of course, which controls epigenetic functions anyway. So unfortunately, it's a rather complicated issue. It applies everywhere to all individuals, all species, but it's the highest complexity we can imagine. It's more simple to work on the genome. But I'm afraid we are in a post-genomic area. We have entered this.
Yeah, absolutely. So the metabolic alteration, and is this known, it's sort of part of the story. Probably more relevant to pediatric will be the underdevelopment of body organs or body for a so how do you see obtain more information on body size, organ function? I see two things happening in my simple thinking. One is a reduction, a modification of enzymatic activity, dyslipidemia, blood pressure, and so on. And the other issue, more relevant to our pediatric, neonatal, perinatal interest, would be the reduction in structure and thus in function of during embryogenesis of organ formation. Do you see this sort of, what is your comment on this? Do we have relevant study that indicates that epigenetic could contribute to organ mass size or hypoplasia? And
certainly, I guess epigenetics control this phenomenon, the growth and the development based on a given genome. And so clearly they have a role. Then the effect on structure is when stressing factors are applied early in development. The effects on function may be the result of later stimuli. And unfortunately, we can have a multitude of phenotypes, as I said, and we have elements of the puzzle, but not all the elements at the moment. And I'm afraid I see these chronic diseases as one disease indeed, because it's the same disease, it's our behavior, our feeding habits, exposure to toxicants, environment we live in, stress, which creates this with different phenotypes according to the behavioral problems we have, based on a genetic predisposition and maybe epigenomic predisposition. But I would see this as a single disease which is avoidable, which is preventable. So this is a major point. And maybe optimizing the entire lifestyle, trying to convince people to educate them and to inform them about optimal lifestyle, what is an optimal environment. It's only a dream to normalize everything, because we don't even know what is normal. The nature is changing, and we are changing the nature by our behaviors, including the climate. And so, but trying to optimize the lifestyles and the exposures would address all types of disease together, likely. We should not aim maybe at preventing only mental diseases, as it has been done in France, or only stroke or diabetes, type 2 diabetes or obesity, but acting on the lifestyle as a whole. And maybe we can have results on all the range of possible diseases, but it's still a dream.
It's very important.
There are several questions that I think we should try to address, as many as we can. But I'd like to start with the first one here. What is the importance of colostrum in developmental programming, especially infants that are exposed to formula instead of colostrum in early postnatal life?
Well, I guess we have, for the moment, little data from this perspective about the colostrum, but it's certainly an extremely important vector of effects. And I would consider that indeed receiving some colostrum early in life is a great advantage, independently on the total breast milk which would be received by the baby. There are many factors at stake. We know today that developmental programming affects the immune system. We know also that the colostrum is very rich in proteins. So the management of proteins controls later the risk of obesity and also of cardiovascular. So we have not characterized this, but it's certainly a very interesting field to be studied. And we know today that even there are microRNAs in breast milk and in the colostrum from the mother. And as during pregnancy, the mother transmits some non-coding RNAs through the placenta to the baby, it is likely that the breast milk, through the breast milk, the mother continues to optimize the epigenome of the baby postnatally, as she made it, as she did before delivery. So it's certainly an extreme importance. Unfortunately, I have little data for the moment on this.
Excellent. There's another question here from Talal Wakar. In fetal growth restricted IUGR babies, what is your take on catch-up growth? Should it be encouraged or avoided?
Well, principal question, but I'm afraid that maybe you and Virgilio could even answer better than me, but I'm afraid we have not enough evidence for the moment to to answer to this question is not a way to cancel it, but that's why I wrote on my slide, optimizing nutrition, starting early, meeting requirements. I'm not sure that we should force a catch-up growth in babies, because for the moment we don't have any evidence of a benefit, and it could be harmful, but we should at least meet the requirements which are known. And the problem, and Chris described this very nicely yesterday, we often induce ourselves an extrauterine growth restriction, which would not be necessary. So let's start with normalizing intakes. And then we have the question of the denominator, which is a key one, but let's say let's try to normalize intakes, and then we may learn more, and obtaining, forcing a catch-up growth. I'm not sure it could work, even if we personalize the approach, taking into account the centile of growth of the fetus in utero, and trying to put again this baby to reach its centile, optimal centile postnatally as a continuum. But I know this is a matter of discussion, and there is a debate. We have very nice growth curves for preterm infants which have been produced, and I'm afraid we need some evidence for intervention. We have learned a lot, but we are missing some trials.
Great. So here's another question from my friend, Professor Ike Kim from South Korea. Thank you for your wonderful lecture. Why and how is the short period of intrauterine and early postnatal period so critical for epigenetic influence? Is there a mechanism suggested or confirmed?
I would say yes, because again, epigenetic changes are part of normal development, and the intensity of epigenetic activity is the highest in life in this time period. So this is maybe a too simple answer, but its first point is that it's not surprising, as the stem cells differentiate thanks to epigenetic mechanisms into a neuron or an epithelial cell, etc. So this phenomenon can be easily unbalanced or altered by an adverse environment. That's how I understand the importance of this period, but maybe it's too simple and there are other explanations.
One more question. I think we're getting close to the end here that we have to stop it. What are the effects of in vitro fertilization on epigenetic or metabolic programming?
Well, both for in vitro fertilization, both the medium of embryo culture and the manipulations of the gametes have been considered to induce some epigenetic changes. And for example, changes in blood pressure or metabolic functions have been observed in the offspring in studies about this artificial reproduction treatments. And the problem is that this was performed with these studies were done with media for culture, for example, and techniques which are ancient now. Then you have a number of confounding factors. First, of course, if you have a single pregnancy, it's completely different than a multiple pregnancy. But if you adjust for these factors, still you have simply what has caused the altered fertility of the parents per se, which could be still at work and influence the outcome of the babies. So I would consider that these babies are doing well globally. Today, we have some follow-up. but a theoretical risk exists, and we need more studies to characterize this.
Well, thank you. Virgilio, do you have anything else to add at this time? I think we're getting close to the end here.
I mean, the message of Umberto on improving lifestyle also, it's well received, and you know, and the idea that is a general, I mean, there is a general hit to the body isn't kind of clear. So, where do you see, Umberto, perhaps as a closing remark, the future next to understanding the mechanism? So, which areas do you think there are some area where a better lifetime would benefit the most, or you see as a whole, as you already pointed out?
Thank you for this really interesting question. I see it as a whole, and I would say, in my opinion, although we are performing a number of manipulations on animals and in vitro, etc., I would say it's time for implementation. Now we know this. And while I was listening to your question, I was just reminding some things that David Barker told me when he saw I was working on epigenetics, and a very, very nice remark. He said to me, well, Umberto, very nice studies, but why, how are you focusing on epigenetics? We know already what happens. Anyway, we don't need epigenetics to act. And he gave the example of the most sophisticated ship, which came out from Southampton. It was a Titanic. What happened to the Titanic while they had, at that time, such a sophisticated ship? And it's true that we need to implement we may find some consequences one day with acting on a specific part of the epigenome, or a drug or a non-coding RNA to be administered to the people, perhaps, and that's why we keep working on this. But we don't need to wait to educate, to sensitize, and to advocate for the environment and lifestyle. And this is a priority. I am very much interested by the experiences which are happening in some countries based on this concept, and let's see if they are positive. But it's a field, it's implementation research. It's a field of research per se, so it's a huge, huge task.
Thank you, Umberto.
Wonderful. Thank you very much. And thanks to the audience for all your attention.
Thank you.
Thank you, Umberto.